gene drive is a self-propagating genetic mechanism by which a desired genetic variant can be spread through a population in a faster and a higher frequency than the Mendelian inheritance.
And so a gene drive is any genetic element that's able to bias its own inheritance. And so normally in most organisms, you have 2 copies of every chromosome, and each parent will contribute one of those chromosomes to its offspring. So that means there's a 50% chance of inheriting either chromosome. And what a gene drive does is it alters that probability of inheritance and is able to get itself into much more than 50% of the offspring.
This is an extraordinary characteristic that makes gene drives a new potential solution for several applications, including but not limited to eradicating or altering disease vectors, such as mosquitoes, controlling invasive species of plants, insects, or mammals, and combating pesticides resistance.
But in terms of trying to recreate a gene drive for our purpose, and what I work on is mosquitoes, and trying to develop a gene drive that can spread a genetic element into a mosquito population that will either cause it to no longer be able to transmit different parasites, or that as it spreads in the population, the gene drive will reduce the capacity of that population to reproduce, and you get suppression of the local mosquito population. And the most successful type of gene drive that can be redesigned for that purpose, at least so far, is based on this copying mechanism. And what this copying mechanism does is it uses a very specific DNA cutting enzyme to cut the DNA of the chromosome that doesn't have the gene drive. And when that chromosome is broken, it needs to repair itself. And the way the cell repair machinery works is to look for an intact chromosome nearby, which is the one that contains the gene drive, and to use that intact chromosome as a template on which to repair and fill in the missing gap of DNA sequence that's created. And when it does that, it's effectively tricked into making a copy of the gene drive on the new chromosome, on the cut chromosome. And therefore, both chromosomes have it. And so regardless of which chromosome is inherited, there's a copy of the gene drive on that chromosome.
Something important to mention is that on its own, CRISPR can edit only the target organism. This means that this organism can pass or might not pass the genetic change to its progeny. This editing technology can revise, remove, and replace DNA in a highly targeted way, allowing us to get to and edit almost any location in the genomes, from yeast and bacteria to mosquitoes and humans. Something really important to mention is that CRISPR doesn't mean gene drive, and gene drive doesn't mean CRISPR. There are several gene drive systems that do not include CRISPR, and many CRISPR applications that not necessarily include a gene drive.
You may have heard of CRISPR. It's a very precise DNA cutting enzyme. We can repurpose that into becoming a gene drive by hooking it up in this type of way. But that doesn't mean that CRISPR itself is a gene drive. If you use the analogy of a vehicle, you have a vehicle that needs to move. You can have different types of motor in that vehicle. It can be horse-powered, it can be pedal-powered, it can be battery-powered, it can be diesel engine. The vehicle in this analogy is the gene drive, and the CRISPR here would be the motor in that vehicle. But there are different types of motors that you could put in there to make the gene drive.